Hello, and welcome back to the Computational Genomics Lab for our last part of ggplot, ggplot 3. We're going to focus more on themes and aesthetics and things you might want to do for your favorite genes. And so let's get started. I've got some packages loaded up there, and then we're going to load in our TPM results. And just so everybody's on the same page, this is O3 plotting ggplot3, our markdown file. Okay, so let's load in our TPM results and our DESeq2 results, which has our res DF filtered, that has our p less than 0.05 and log two-fold change greater than absolute value 1. So we're going to look at facets, like how to make a bunch of plots using the same data, but make individual plots for each gene. So let's get started with that by just making a list of your favorite genes. I picked these by looking through the data that I think they're interesting, but my actual favorite gene of all time is fire. I put that one in there for the heck of it. And so we've made an object called genes, which is just simply our genes, and we're going to do some tidyverse to say, go into our filtered res df here, pipe that into the filter function, and gene name in genes. So basically it's going to go find these genes in our big res df and only grab out those genes. So we can plot those. Okay, cool. So we have our gene values here. So we see we have these 11 genes. That means that it occurred in more than one time point. So some of these genes are changing in more than one time point because we only put in 6 genes, but we got 11 rows. So that means 5 of them had at least, more than one value. Okay, or changed in more than one time point. Okay, so let's plot this. So we're going to take our gene values, that little mini data frame we have now. We'll make the x-axis log two-fold change. Y is p adjusted. We're going to facet grid these by gene name. So we're going to make a plot for each gene name. We went over facet before, but it needs a tilde, and you can do it against another column in your data frame, or just say here with the dot. And we're going to make a geom point line here. Okay, so this is kind of busy because we made a plot for each gene, but now what we can see is that this PGK gene is very highly up and has 3 data points, which means it changed in more than one. And we didn't grab fire because fire didn't make our filtered list. So we only got 5 genes. If it's not in that list, it's not going to be able to plot it. But we can see these 5 genes are the ones that have that very low, they decrease a lot, and this one increases a lot, and this one barely changes at all. That way you can sort of make a plot of your favorite genes by making an object, filtering them, and going from there. Okay, so now I believe this is exactly the same thing. Oh no, instead of facet grid, we're going to compare it to facet wrap. Okay, so that's the 2 differences that we're trying to learn. And technically I tend to just run them and see what happens and which ones I like better. And so I think this one looks a little better. We have each data point here of where it was significant. So this was significant in 3 time points, and this one was in 4, 1, 1, and 1. Okay, very interesting. So now we can see that we can make a lot of plots for our favorite genes, and it'll go from there. And even if the gene didn't make it into your data frame, it'll still plot the ones it can find. Okay, so let's do 2 facets now, where we did the before facet wrap with gene name tilde dot. Now we're going to do it to result name. So now we'll know which time change, or which time point it changed in. So we're going to take our gene values, which is our little mini data frame, same x and y axes, facet wrap to 2 functions, and geom point. Okay, let's see how that changes. Okay, lots of information here. So now what we're seeing is that it's labeling them by the time point. So what we can see is NeuroD had 2 changes. One, both of them pretty close to 0 or twofold change. Where's the one that has the most? Okay, so this one also has 3, and so we can see that it's changing in the 12 versus 0, 24, and 48 significantly, and we can see it moving slightly to the right, and that's probably why it was significant. So now you can have sort of plot these out to sort of clearly see each dot by itself. Okay. And then now we will do, these are the same genes. We're not adding fire anymore. We took fire out. So we're making just another little list of genes, and we're doing the same thing here. We're piping the ResDF to filter just to these gene names. Okay, and we rewrote over the previous genes. And now we're going to do the same plot, but we're going to do, instead of facet wrap to 2 things, we're going to look at facet grid. Again, I tend to just try both of them and see which one looks better. And let's see what we find out here. Okay, so I think this looks a little better. So now we have our genes are all on the same, each one is a row, and that's what the grid did. So now we can see that this one only changed in this time point. This one changed across all 4 time points, and this one again was across the first 3. So we can start to get some more granular detail of when and how much the gene changed due to DOX exposure. Okay, so now we're going to go and modify this to the way we want it to look. So we're going to continue with facet wrap here, and same X and Y axes, and we're going to wrap the gene name as we did before, just to itself, but we're going to say that we want 2 rows and 3 columns, so we can specify what this is going to look like and tailor it to what we want. And it looks fairly similar to what we saw before, but just so you know that you can actually tell it to organize it the way you want. And again, you can always export any of these figures, put them into Illustrator, and change colors and things that way. Okay, so now we are going to do, we can see that in this plot, all the scales are the same. So even though we have 3 different genes here, it's the same scale for, all 5 genes here. So what we can do is say scales free, and then it'll scale each box to its own value. So you kind of get it all in relative to that one gene and that time point. So same thing here, gene values is what we're plotting, same X and Y, same facet wrap to gene name, same rows and columns, but now we're going to say scales equals free. And you can look in ggplot under facet wrap for other parameters you might find interesting. And again, just the plus here is like a pipe. If you forget one, it'll stop where the last one was. Okay, let's see what happened here. Cool, so now we can see that the scales are very different. So we can see the P adjusted on this one is super significant. It doesn't even start until before that one began or ended. So here we can see now that this gene is highly significant, and even its biggest number is still 1 by 10 to the negative 3. And same with NeuroD, and PGK is just kind of on the edge, and same with GM163. So now each scale was made relative to the data for each gene. So that's really powerful that ggplot can do all this. Okay, so now let's say, again, I don't think you're going to ever want to do this, but just to sort of complete the layers of ggplot is we're going to look at the coordinate layer. So everything we've done so far is in Cartesian space, X and Y, bar plots with just X axes. And so now we're going to kind of go into a different dimensional space where we can plot this. So let's take our filtered ResDF. We'll say the x-axis equals the result name. So we'll have 4 bars here with geom bar. But now we're going to add coordinate layer as polar. So it's a lot of math that I don't understand, but basically turns your data into a circle, and now you can plot it to see kind of what we saw before with the bar plot, that most of our genes are changing in the 24 to 48 time points. So if you just want to switch it up and make your bar chart sort of circular, coordinate space is, or polar space is for you. So now we can also tell it the theta is y, or the y-axis is the theta. So this is all the same code here. We have the AES is result name, and then we're adding the coordinate layer, but now we're putting the polar layer, but we're adding in this parameter y. Again, you would get that from the manual by doing question mark. And so now we can see the size of each bar plot within, so again, 12 hours and 96 are not very many genes changing. Majority of the genes are changing in 48 and a little less than 24. So this is another way of visualizing it, but it's really a bar plot in polar coordinates. Okay, theme. This is one of my favorite parts of ggplot, is you can really make your favorite colors. Like if you want a clear background or you want lines in your background, you can decide what you want, and then you can make a theme for that. But there's also themes built into ggplot. So like this one is theme gray. So here we're going to do a density plot like we've done before. So we take our x-axis as log fold change. We make a density plot with alpha so that they don't overlap as well. The opacity is changed. We're going to put in these two colors. Just pick them randomly, and then we're going to do theme gray, which is in the ggplot package, and it knows what kind of aesthetics you're going to want, how big the title should be, where it should be placed, etc. So you don't have to do that every single time. And now we're going to add labels. So we're going to say the X label is the expression log fold to change. That's the x-axis. The Y label is density, and GG title is log 2 fold change, and the subtitle genes regulated by Dachshund time. So now it's going to look more like an official figure that you could present in lab meeting, and we can see that the log fold 2 changes are mostly around 1 and -1, but then there's data all the way out to -20. And again, we might have slightly more genes going up in here. Okay, so it's starting to look more professional, and now we're going to make a monster plot with all the things we've done and sort of summarize what we've been working on. We're going to use that broom function to get our linear model data plotted. Okay, so at first we're looking very similar here. AES, p-value, and p-adjusted. So that is going to default to x and y, even though we didn't say x equals. I got a little lazy there, but you've seen that enough times up here that you put the x equals in there, but it'll default to x and y. So we're going to make a point plot, and we're going to make a smooth linear model. We're not going to show the standard error, so that's another parameter. We could change that to true, and I encourage you to go do that. So we're going to say geom smooth is the next layer. The method is a linear model, and don't show the standard error. And now we're going to have some geom text. It's just a different way of making those labels. And so we can say x equals the mean filtered res p-value. So we're going to get the mean p-value from our all of our data. And max, the y-axis is going to be the max p-adjusted here. And then we're going to label, this is that paste that is like print or cat, y equals. And now we're going to pull out the coefficients and the data from the linear model like we did before and put it in our plot. So we're going to say take the coefficient of the linear model and round it. So we're going to round it so it doesn't have a really long number. And we're going to take filtered res DFP value and compare it to the p-adjusted, so that's going to be our linear model is p-value versus p-adjusted. Again, the order does matter. And we are going to take our filtered res p-value out of that. And then this was that second plot, second thing we're extracting from the linear model. If you go back one class when we did that, you can see the data frame that's produced, and we're in ggplot doing that same code we did in just R. And again, we're going to round the coefficient for the x-axis in here, and again grab now the first and second. You can go look through these, but these are going to be the same every time you run the linear model if you just want to memorize them. We're going to make the colors red and a size, and then a title and a subtitle. Okay, so that's a lot you can see you can do with ggplot, and you're running code inside of ggplot, and we get this nice plot here where we can see we can make a function of y is the function of 0.2x plus 0, so the intercept is at 0. Here's our linear model. We have a title there, and we have the values of our linear model, which I think this could be cleaner. There's a way you can just highlight your code and it will clean it up for you. It's Command-Shift-A. Command-Shift-I. Nope. There's a way to clean that up, but that's not the cleanest looking version of ggplot. Okay, so you can make custom themes. We've made a custom theme for this class. So the same way we've sourced functions before up top in our sort of starting plot panel. I've made I'm going to make sure I am set to this working directory. Oh, I'm in the wrong one. Look at that. So I want to go to 07, visualization of figures. Click in there, set as working directory. And we've made this theme paper white. A former student made this, Michael Smulligan. And so we're going to do the same plot we just made, but using this theme that is custom made. And it's in our utility folder, which is a couple directories up. That's a common place to put R scripts and different plotting themes you might want to call up. Okay, so now it has all these things we can use in our plot that we made custom. And voila, it looks pretty similar, but it's a little cleaner, paper white, so there's no lines. But we got the same sort of output here. This is the linear model created, and then these are the coefficients. You could also grab these in a different order, but you just want to look at that LM object. What are the objects you want out of it, and then use Broom to plot them. Okay, so now what we're going to do is just do a facet grid by gene name. So we're going to take our gene values, just those few genes we made at the top, plot the log fold change versus p-adjusted, theme paper white, and we're going to call out this color panel within there. So in our custom one, there's a bunch of different color palettes called with these names. I'm going to do a facet grid by gene name and make a dot plot. Okay, so now if we zoom in, we can see it's kind of similar to what we saw before, but there's way less lines. It might actually be easier to read with the lines, but I like to tend to have these clean plots here. Again, we could have done scales free to make this a little more readable. Okay, so there's a lot in here, and as I mentioned in the last class, just pick and choose what things you kind of like. And these are really the basics to get you to start Googling. You can also use ChatGPT, say, hey, I want this figure to look like this with these values, and you'll now know the logic of where to put the different layers, and you can run functions in the aesthetics layer and do lots of code. So ggplot is super powerful. There's more on custom themes here. And next we're going to be starting to make some plots with our TPM data. And this was all DEC, like log fold to single value things, and now we're going to start plotting our TPMs across time for these genes and go from there and make more figures. Okay, I'll see you next time.